Hello everyone. Our topic is estimation of hardness of water by EDTA method. It is well known as EDTA method, but actual name is complexometric method. Before going to explain about estimation of hardness of water, first we need to understand what is the reason for hardness. Hardness is mainly due to the presence of dissolved salts of calcium and magnesium. So what are the hardness producing metals? Calcium and magnesium. Due to the, if water consists of salts of calcium and magnesium metals, that water is considered as hard water. Here our concept is what? Our aim is to estimate the hardness of water by EDTA method. Okay, why this method is called as EDTA method? What is complexometric method? First look into the definition of complexometric method. The method in which formation of stable complex takes place in between hardness causing metals and complexing ligand, that is EDTA, is known as complexometric method. I will simplify this. Here, why this method is called as complexometric method means, complexometric method is a method in which whenever we add complexing agent or complexing ligand to hard water, this complexing ligand captured the hardness causing metal ions, that is calcium magnesium. Whenever we add EDTA, that is nothing but complexing ligand. EDTA is what? EDTA is a complexing ligand or complexing ligand. Whenever we add this complexing ligand or complexing ligand, that is EDTA, to add to water, hard water, that will capture the hardness causing metal ions and it will form one stable complex with these metal ions. That is why it is called as complexometric method. Okay, so here it is a water. Consider it is a water. This water consists of calcium, magnesium. Whenever this water consists of calcium, magnesium, whenever we add EDTA to this water, this EDTA, observe here, it is the calcium and hard water which consists of calcium and magnesium to this hard water. Whenever we add EDTA, this EDTA captures the hardness causing metal ions and it will form one stable complex. So here in this method, stable complexes form. That is why it is called as complexometric method. Here complexing agent is what? Or complexing ligand or ligand is what? EDTA. That is why it is also called as EDTA method. Next, what is EDTA? EDTA stands for ethylene diamine tetraacetic acid. It is a complexing ligand. Observe here, E stands for ethylene, D stands for diamine, T stands for tetra, A stands for acetic acid. Look into the structure of EDTA. Here 2 carbons indicates ethylene, 2 nitrogens indicates diamine. 2 means diamine. 2 nitrogens indicates diamine. Ethylene, diamine, tetra. 1, 2, 3, 4. Acetic acid groups are 4. That is why it is called as tetraacetic acid. This is the overall structure of EDTA. Ethylene, diamine, tetra. 1, 2, 3, 4 acetic acid groups. That is why it is the structure of EDTA. That is ethylene diamine tetraacetic acid. Here I said we are adding EDTA to water. Which form of EDTA we are adding? Generally we used EDTA which consists of disodiums. That means 2 hydrogens. Here 4, in acetic acid 4 hydrogens are there. No? So that among that 4 hydrogens, 2 hydrogens are replaced by sodium. Whenever we replace, observe the first structure and second structure, everything is same. Only the difference in 2 hydrogens. Here instead of hydrogen, that means first hydrogen is replaced by 1 mole of sodium, second hydrogen is replaced by 1 mole of sodium. So how many sodiums are there? 2. That is why it is called as disodium. The remaining structure resembles what? EDTA. So this is the overall structure of what? Disodium salt of EDTA. Whenever we add this EDTA to water, it is a titration process. It is a titration process means what? It is a practical. It is important for theory as well as it is one of the practical according to R22 Engineering Chemistry Lab syllabus. It is one of the practical according to EDTA, according to that method theory. It is the most important long answer question. Look into the theory of this. So what is this titration process? What is actually happening? What are the chemicals required? What are the apparatus required? And what are the steps involved in this method? How to calculate or how to estimate amount of hardness present in this hard water. In the hard water by using this EDTA method. Just now I said it is a titration process. For titration, first we need to collect the chemicals. What are the chemicals we need? Our aim to calculate hardness. So if our aim to calculate hardness, our first requirement is what? Hard water. Second one is what? It is calculated by using EDTA. So EDTA is second chemical. Third chemical is one indicator. Here hard water is colorless, EDTA also colorless. Whenever we add EDTA to hard water, how can we identify whether the color reaction is completed or not? To identify the completion of reaction or end point of the titration, we should add one indicator. Here we are using one indicator, that is EBT. What is EBT? EBT stands for Eriochrome black tea. It is an indicator which is used in this EDTA method or complexometric method. This EBT indicator is in blue color. You should remember the colors here. Colors are very important. What is EBT? EBT is an indicator. EBT stands for Eriochrome black tea. This EBT indicator is blue in color. And one more chemical we are using. What is that one more chemical? Means here we are performing the experiment with water. To maintain the pH of water at 9 to 10, we have to add buffer solution. Which buffer solution we are adding? Ammonical buffer solution we are adding. Ammonical buffer solution means ammonium hydroxide and ammonium chloride. So chemicals required for this EDTA method is, first one is hard water, second one is EDTA, third one is EBT indicator, fourth one is ammonical buffer solution. By the way, it is the structure of EBT, that is indicator. It is the structure of the indicator. Look into the principle. If you understand the principle, you can understand the procedure very easily. Look into the principle. What we have to do in this experiment means, first we have to take the hard water. In that hard water, we should take hard water in a conical flask. Just assume it is a conical flask. In this conical flask, first we have to take what? Hard water. To this hard water, what we have to add? We have to add EBT indicator and buffer solution. And take the EDTA solution in burette. It is what? EDTA. It is EDTA which is taken in burette. Observe here, what are the chemicals? Hard water, EDTA, EBT, buffer solution. Here we are taking hard water, EBT and buffer solution in conical flask, and we took EDTA solution in burette. Okay. Here water is colorless. Buffer solution also colorless. But whenever we add EBT indicator to hard water, the solution turns into wine red color. This process is explained by this reaction. Observe here, calcium. Here calcium indicates what? Hard water. Just now I said hardness is mainly due to the presence of what? Calcium and magnesium metal. So here hard water is represented with calcium. To this calcium or hard water, we are adding what? EBT indicator and buffer solution. EBT indicator is blue in color. EBT indicator. What is the color of EBT indicator? EBT indicator is in blue color. Whenever this EBT indicator is added to hard water in presence of buffer solution, it will turns into wine red color. Why it turns into wine red color? Due to the formation of unstable complex. What is that unstable complex? Calcium EBT is an unstable complex. Not only calcium, magnesium also will form an unstable complex because hardness is mainly due to the presence of calcium and magnesium metals. Look into the reaction of magnesium. Magnesium also react with EBT indicator. You should remember here, magnesium, whether magnesium or calcium indicates what? Hard water. So to this hard water, initially we are adding EBT indicator and buffer solution. Whenever we add EBT indicator and buffer solution to hard water, it will turns into wine red color. Colors are very important. 99% of the students will commit mistake while writing the color of the solutions. So here, whenever we add EBT indicator and buffer solution to hard water, initially it will turns into wine red color. You should remember one important point. Till now we didn't add any EDTA solution because EDTA is taken in burette. These all are taken in conical flask. Okay, whenever the solution turns into wine red color due to the formation of unstable complex, that may be calcium EBT or magnesium EBT. So to this solution we have to add EDTA. Observe here, this is the product to this product. In first reaction, what are the products? Calcium EBT and magnesium EBT are the products. So if you understand this diagram, you can understand the reactions very easily. Here we added EBT indicator and buffer solution to hard water. So whenever we add EBT indicator and buffer solution to hard water, it will turns into which color? Wine red color. To this wine red color, we are adding what? EDTA. Drop wise we are adding EDTA. Whenever we add EDTA solution to wine red color is nothing but what? Wine red color is nothing but calcium EBT and magnesium EBT, which is an unstable complex. To this wine red color, we are adding what? To this wine red color, we are adding EDTA drop wise. Look into that reaction. Here also we are adding EDTA by titration process. It will turns into what? Here EDTA is a complexing ligand. Just now I said EDTA is a complexing ligand. Complexing ligand means what? What is the nature of EDTA? Whenever we add EDTA to this wine red color, it will capture the metal ion from the unstable complex and it will form one stable complex with the calcium and magnesium metal ions. What is the nature of that? That is a stable complex. And one more important point you should remember here. This EBT is replaced by EDTA and EBT is released. In first reaction we are adding EBT. In next reaction EBT is released. What is the actual color of EBT? EBT color is blue. Whenever it combined with calcium and magnesium, it will turns into wine red color. Whenever we add EDTA to wine red color, it will replaced by, that means EBT indicator is replaced by EDTA and release of EBT indicator happen. We know the color of EBT indicator, that is blue. But actual product, that is a stable complex which is formed in between calcium EDTA or magnesium EDTA, actually doesn't have color. So here main product doesn't have color. But due to the release of an indicator, that is EBT, we are observing blue color is the end point of the titration. So first what we have to do, first understand this diagram, then you can understand the reactions very easily. Take one conical flask. In that conical flask we have to take hard water. To that hard water add 2 to 3 drops of EBT indicator and buffer solution. Here we are adding buffer solution to maintain the pH of water at 9 to 10. Whenever we add EBT indicator and buffer solution to hard water, the whole solution in conical flask is turns into wine red color. To this wine red color, we have to add EDTA through titration. Formation of this wine red color is explained by first reaction and second reaction. To this wine red color, we are adding EDTA. What is actually happening whenever we add EDTA is explained by third reaction and fourth reaction. So overall, this is the procedure. It is the principle. Look into the procedure. Procedure means it is related to practical terms. Take a burette and fill the burette with EDTA solution. Next, 20 ml of hot water sample taken in a conical flask. These are all points just now I explained through this diagram. So it is the simple form to remember. This is the theoretical form to explain in examination. So 20 ml of hard water sample is taken in a conical flask. To that hard water we should add 1 drop of EBT indicator and 5 ml of ammonical buffer solution. The solution turns into wine red color. The wine red color solution is titrated against EDTA until the color of the solution turns into blue color. Whenever it turns into blue color, we should stop the addition and note down the burette reading. By using burette reading and remaining values, we can calculate the hardness of water very easily. Look into the calculation part. To calculate the total hardness of water, here we have to take. So here two methods are there. That means two steps are there. First step is total hardness. Second step is permanent hardness. Permanent hardness is nothing but the hardness which is separated from temporary hardness. If you remove the temporary hardness from total hardness, the remaining water is considered as permanent hard water. First we have to take total hard water. Total hard water is nothing but tap water. In this step we took tap water. So formula is M1V1 equals to M2V2. M1 is the molarity of sample hard water. V1 is the molarity volume of hard water. V2 is volume of EDTA solution. M2 is molarity of EDTA solution. Look into the formula. Here hard water sample concentration we don't know. That is why it is question mark. Volume of hard water. How much we taken in conical flask? We took 20 ml. That is why V1 is 20 ml. M2 molarity is EDTA concentration, that is 0.01 m. Next, volume of EDTA we will get from the burette reading at the end point. So among four terms we know the M2 value, V2 value, V1 value. So if you know the three values, we can identify the fourth one very easily by using the formula M1 equals to M2 V2 by V1. It is the molarity of tap water. To calculate total hardness, we should multiply the tap water molarity, that is M1 with 100 and 1000. We will get the total hardness of water in terms of ppm. So this is about total hardness. Next, to calculate the permanent hardness, instead of tap water, we have to use permanent hard water. If we use permanent hard water, we will get the permanent hardness. So we should multiply the permanent hardness molarity with 100 and 1000. We can get the permanent hardness. We know the formula. Total hardness equals to temporary hardness plus permanent hardness. One step we can calculate total hardness. One step we can calculate permanent hardness. If you know the two values, we can calculate temporary hardness very easily by using the formula temporary hardness equals to total hardness minus permanent hardness. How to calculate permanent hardness means I'll explain how to calculate permanent hardness now. To calculate permanent hardness, first we have to take 250 ml of water. First we have to take 250 ml of tap water. Okay, we should heat the tap water for some time until its volume reduces to 50 ml. We took 250 ml water. We need to heat the water until its volume reduces to 50 ml. Whenever the volume of water reduces to 50 ml, we observe there is a formation of one type of precipitate in this. Observe here, it is the total hardness containing water. Total hardness means what, ma? Total hardness means temporary hardness plus permanent hardness. First we are taking which type of water? Total hardness containing water. We are heating the total hardness containing water to 50 ml hard water until its volume reduces to 50 ml. So this is the 50 ml water. Whenever we observe the 50 ml water, here 250 ml water doesn't contain any dissolved salt settled at the bottom. Whenever its volume reduces to 50 ml after heating, we observe there is this lot of impurities which are settled at the bottom of the beaker. These impurities are called as temporary hardness causing dissolved salt. which we can remove easily by filtration process. So we should filter that water. Whenever we filter that water by using funnel, the impurities are dissolved salts or precipitates are settled at the filter paper. We will get the remaining water. Observe here, initially we are taking total water. Total hard water is nothing but what? The water which consists of temporary hardness and permanent hardness. We know temporary hardness we can remove easily by simple boiling followed by filtration. But permanent hardness we cannot remove easily by simple boiling process. So to calculate the permanent hardness, we have to remove the temporary hardness from the total hardness. To remove the temporary hardness from total hardness or to get the permanent hard water, first we have to take the total hard water. We should boil the total hard water. By that we will get the temporary hardness. How to remove the temporary hardness? By filtration process we can remove the temporary hardness. Finally, this is the temporary hardness. We are getting which type of water? Permanent hard water. From this permanent hard water, we should take 20 ml of permanent hard water in a conical flask. Add EBT indicator and buffer solution. Same EBT solution and same buffer solution we have to take. In first step, second step, what is the difference is, in first step, our aim to calculate total hardness. To calculate total hardness, we used tap water. To calculate permanent hardness, we are using boiled water or permanent hard water. Same procedure. So we will get the answer. So this is about EDTA method. By using this EDTA method, we can calculate the total hardness, temporary hardness and permanent hardness of the water.